All right, I think we're live. Welcome, Julijana. Welcome everybody else to the Theory Forum at Worldwide Neuro. My name is Tim Vogels. I'm a professor at the Institute of Science and Technology in Austria, and I'm happy to welcome Julijana Gjorgjieva to our seminar series today. Julijana got her PhD at the University of Cambridge in the UK, and already during her PhD, I think, started commuting to the University of Washington, working both in Cambridge and on the West Coast in the United States. And from there, moved on to the East Coast, to Harvard University, where she worked with Chaim Sopolinsky, and later in the same city with Gina Giorgiano and Eve Marder at Brandeis University, and finally became a group leader in 2016, such that she is now affiliated with both the MPI in Frankfurt, as well as the Technical University in Munich, where she's now, I suppose, an associate professor, a W2 professor in German lingo. And today, Julijana is going to tell us about how synapses organize themselves by way of synaptic plasticity on cortical dendrites. Generally speaking, synaptic plasticity is something that Julijana is quite well known for now, and I'm excited to hear all about it. So without further ado, Julijana, the screen is yours.
Great. Thank you very much, Tim, for the kind introduction and for the invitation to join the Worldwide Neuro Seminar Series, which is now indeed world famous. I've been following this series since the start of the pandemic, of course, and I'm really happy to finally be invited and to take part and to tell you a little bit about a recent work. So before I start telling you about how synaptic inputs become organized on cortical dendrites and the activity-dependent mechanisms that drive this organization, because I don't really have a chance to give you an overview of all the different kinds of things that we work in my group, I added this first slide to just summarize the different types of approaches and questions that we try to address in my group. So we're a group that purely uses theoretical and computational approaches to understand how organization and computation emerge in neural circuits in the brain, and to address these questions, we use two different types of approaches. The first one is what I call mechanistic or bottom-up approaches, where we study neural circuit computation and function by really focusing on the individual components that make up the circuit. So these are the single neurons, as well as the connections between the neurons. So in particular, we're interested in sort of how changes in ion channel properties and changes in the strength of the synaptic connections through mechanisms of activity-dependent plasticity shape, structure, and function in these neural circuits. And one of the applications of these types of approaches is especially in the context of development. So how neural circuits become organized in very, very early stages of postnatal development, for instance, in the context of the visual system before eye opening. At the same time, we also use so-called normative or top-down approaches in my group. So here we try to get at the question of how cell types and computations emerge, not by focusing on individual components, namely the cells and the synaptic connections, but on kind of more overarching principles that might underlie this organization. For example, maybe optimizing information transfer. And one powerful framework that we have been using in the past few years has been the so-called efficient coding framework that postulates that maybe cell types have come about because as a population, they efficiently encode a given sensory stimulus. And more recently, my group has started to go beyond just activity and dynamics in neural circuits, but to go sort of from circuits all the way to behavior. And in this case, actually, in the case of all the approaches, we work very, very strongly and in collaboration with experimental colleagues who collect the data to help us. We help them interpret the data and then build models that are very strongly inspired and driven by this data. And this is especially the case in this last approach that's kind of more recent for my group, where we try to go from circuits to behavior. And so here we work with model systems like Drosophila, the fruit-fly Drosophila in mice, and we look at different types of behaviors, such as state-dependent behavior. So what happens when animals are hungry or starved and hungry or satiated. And we investigate behaviors like locomotion. So what we can see here with larvae of Drosophila. All right, but what I will tell you today is one recent project that we have addressed is with one of my PhD students that falls in the broad framework of these mechanistic or bottom-up approaches, where we're interested in how organization emerges actually at the subcellular level, and how this comes about during very early postnatal development through activity-dependent mechanisms of synaptic plasticity. So very generally, in the context of development, we're interested in how basically the brain of a developing fetus acquires all the important connections and properties that it needs to function as an adult. So how does the brain of a baby go from basically this to this, to being able to execute all these fancy behaviors and tasks? For instance, play with a cell phone, with a smartphone. Of course, this is a process that involves a large number of mechanisms, and they can very broadly be divided into these two different classes. So activity-independent mechanisms that underlie the interaction of different sets of genes and molecules. So these are not the ones I will be describing today. And the second are activity-dependent mechanisms that rely on specific patterns or aspects of neural activity to basically determine or to drive the emergence of this non-random structure and eventually function in these circuits as they develop towards adulthood. And so typically when we think about neural activity, we think about neural activity as such as generated, especially when we think about circuits in a sensory cortex, we think about activity that's generated by the sensory organs. So this is sensory evoked activity. But as I mentioned, in this context, we think about development, so very early stages of postnatal development, when actually many of the sensory organs, such as the eyes, the ears, the nose, are immature, and they cannot basically process, or they're not even capable of processing sensory information as they are in adulthood. So this, of course, raises the natural question, well, if they cannot process sensory information, then where does activity come from? And the answer is, well, it is spontaneously or endogenously generated in the circuits themselves. And so here I like to show this example of such spontaneous activity generated in a 3-week post-conceptional age, so preterm infant, generated in the somatosensory cortex. So these are EEG recordings. So at the top, you see basically trajectories of movement. So you see there's basically no movement in this baby, but at the bottom, you see electrophysiological signatures of activity that is actually completely spontaneously generated in the absence of movement. So these are these so-called delta brush oscillations that are highlighted here in the gray boxes. And so indeed, we have these highly non-random patterns of spontaneous activity that we know now have a very important role in driving different aspects of organization in a large number of neural circuits. And so today we're going to be talking about the visual system. So as I already mentioned here, spontaneous activity is generated in the first 2 postnatal weeks after the animal is born. So eye-opening in the rodents. So we're going to be using, we're inspiring our work based on recordings and recent experiments done in the rodent visual system, so mouse and rat. So here eye-opening is at P15, and spontaneous activity is basically generated for the entire period prior to eye-opening. So we're asking questions about how spontaneous activity can actually refine circuit connectivity. But of course, we're interested in what happens after eye-opening. So again, sort of somewhat early stages of development, but after eye-opening, where we're interested in how these now newly established circuits by these non-random patterns of spontaneous activity are maintained, especially in the presence of different kinds of perturbations. So for example, one of the some of the projects they address in my group is paradigms like monocular deprivation, where you can close one of the eyes of the animal, so you disrupt visual input to the visual cortex and to the rest of the brain, and you basically investigate what happens to the network dynamics. and study mechanisms that maintain normal function despite this disruption of sensory experience. So as I mentioned, we do this in the visual system of rodents. So let me just show you what spontaneous activity looks like in the visual system. So here, spontaneous activity is propagated throughout the entire visual system. So it begins with the first organ that processes vision, and that is the retina. So what we see here in the top video is activity that is generated in the closed eye. So remember here, the eyes are closed. This is taken around postnatal day 7 to 10. So 1 week after the animal is born. And so what we see here in this calcium imaging recording done by my collaborators in the lab of Christian Lohmann in Amsterdam is very beautifully patterned spontaneous activity that has been known now for over 30 years that propagates in the form of retinal waves. So these are known as retinal waves. This spontaneous activity propagates throughout the entire visual system. So I'm not showing here other circuits in the brain. I'm just showing the eyes and the visual cortex. So of course it's relayed through the LGN, which is the visual area of the thalamus. It goes to the visual cortex. And in the visual cortex, it actually manifests itself with slightly different patterns. So they're not as beautifully spatiotemporally patterned as the waves in the retina, but they sort of tend to have these more modular patterns. So you have these spatial locations or areas where you have these sort of bouts or these modes of activity. Of course, the spatial scales of these two different videos are different. At the top, we have about 0.5 millimeter, and here we have several hundred microns. But still, these aspects of the activity where in the retina we have waves and in the cortex we have more modular activity remain. And so, this activity, I mentioned here the visual system because that's what I want to talk to you about in the next 40 minutes. But this spontaneous activity is seen literally in all the circuits that you could think of in the brain in development, in hippocampus, in the spinal cord, in cerebellum. It's literally seen everywhere. And now, through many experiments done over the years by different labs, we know that if you block this activity or if you disrupt certain patterns of this activity, we know that you can affect different aspects of organization of connectivity in downstream areas. And so we know that the spontaneous activity indeed has a specific, very specific and distinct function and role in circuit development. Okay, so in my group, we have several projects and have been looking at this for the last few years to understand what exactly it is about this spontaneous activity and the patterns in the spontaneous activity that might drive circuit refinements in a way that sort of makes these circuits transition from being, inappropriately connected early in development to being appropriately connected later on and capable of to do all these tasks that we can do. Most of the time, we actually address this at the circuit level. So, we look at data that's like the one I show here on the right, say 2-photon calcium imaging recordings, where we look at the individual activity at the level of the single cells. And so we look at network features that change as a function of development or maybe as a function of a specific type of perturbation. And we build models to understand mechanistically what are the say plasticity mechanisms that underlie these changes. But more recently, we have been looking at how this spontaneous activity drives circuit connectivity beyond the circuit level, namely at a much smaller scale, so at the level of the individual synapses that impinge on synaptic inputs that impinge on a given neuron. So here we take a pyramidal neuron in the cortex, in the visual cortex, and we were particularly inspired by some recent recordings. So as early, or I should say as late as 2011. So these were really the first recordings that were done now less than 10 years ago that first characterized the organization of synaptic inputs on these cortical pyramidal neurons. And so, interestingly, in these first experiments, so the first study here that I cite was done in the hippocampus and in the visual cortex, and the second study was done in the somatosensory cortex. Two different labs used the calcium imaging to investigate basically the correlated activity of individual synaptic inputs on individual dendritic branches of these neurons, either in the two sensory cortices or in the hippocampus. And so what they did, so this is actually, this is a figure that I have taken from this first study. What they did is they investigated the joint activity of these synaptic inputs, so the coactivity of synaptic inputs as a function of their distance. So they mapped the synaptic inputs on these individual branches, and then they plotted the coactivation, so the percent of time that these neurons coactivate together as a function of how far these synaptic inputs are from each other in microns. What these studies were able to find was a so-called, we call it a fine-scale connectivity rule, which basically means or shows that synapses that are nearby each other, so within 15 microns or 12 microns of each other, are very strongly coactivated, and synapses that are far away from each other, so more than 15 microns apart, basically are not coactivated more often than chance. So we call this a fine-scale connectivity rule, or basically a functional synaptic clustering, because it tells us that synapses that are coactive are nearby each other, namely synapses that are coactive are clustered. And we know, so this was done in very early development, and we know that this is imprinted by patterns of spontaneous activity, because several different experiments that blocked spontaneous activity revealed that this particular relationship, this fine-scale connectivity rule, was eliminated. So actually, you block spontaneous activity, you don't see this synaptic clustering. Okay, so with my student Jan Kirchner, a very talented PhD student in my group, we wanted to address the question of whether these non-random spontaneous activity patterns that I described to you, that propagate throughout the entire visual system, whether they could contain sufficient information to imprint this fine-scale organization of dendritic synapses into these clusters that were identified in the different sensory cortices and the hippocampus. And so, again, we thought a lot about the different kinds of activity-dependent mechanisms that could drive this process, and we settled on a very, again, recently identified mechanism based, we call it a biophysical mechanism because it's based on a particular set of molecules called neurotrophins that were identified to be specifically implicated in the synaptic clustering that emerges in the development in the hippocampus. So what are neurotrophins? So these are a family of proteins that actually govern the growth, and so the birth and the growth of synapses, and also their synaptic pruning. So they're responsible both for the proliferation of new synapses and their eventual growth, but also for their eventual depression and elimination. And so most of you might be familiar with some kind of neurotrophins. You probably have heard of the brain-derived neurotrophic factor, or BDNF, which is a neurotrophin that is present in the mature, so in the adult nervous system. And this particular neurotrophin is known to bind to a particular receptor, namely the TrkB receptor, and it's supposed to be involved in sort of positive processes. So processes like the proliferation of new synapses, the potentiation of existing synapses, so sort of positive growth processes. However, the Lohmann lab in Amsterdam recently identified that a different type of protein, namely different type of neurotrophin, that's a complementary to BDNF, known as proBDNF, which binds to a different type of receptor, P75 neurotrophic receptor, is actually responsible for the opposite action. So it's responsible for the depression and eventually the elimination of synaptic inputs. And actually, even before they discovered this in the developing hippocampus, this kind of idea that you might have these two opposing neurotrophins, one BDNF and the other one proBDNF, has been known before and has been characterized, for example, in the neuromuscular junction. It has been known as the so-called yin-yang hypothesis of neurotrophic action, that you have one positive and one negative neurotrophin that tend to play opposite each other. But it turns out that in development, you actually have a little bit more of this proBDNF molecule. And so this suggests that you might have significantly more depression and elimination of synaptic inputs. So this is indeed the case on average, but there is another molecule, in this case, a matrix metalloproteinase, MMP9. This is a very well-studied molecule that is activity-dependent, released in an activity-dependent fashion, and is actually known to convert this depressing proBDNF into the potentiating BDNF, and therefore can serve as a switch between this positive and negative action played by these two neurotrophic factors. And so the interaction between these two opposing neurotrophins through MMP9 was recently uncovered experimentally. And so we wanted to see whether we could formalize a mathematical model that uses these particular neurotrophins, and then apply this model in the context of the problem that I described to you so far, namely the organization of these synaptic inputs into clusters, especially in the context of development when they're subjected or they're experiencing spontaneous activity. Okay, so let me just schematically illustrate the model that we conceptualized in this case. So in the corner here, we have this interaction between proBDNF and BDNF, where proBDNF, which is this depressing molecule, is converted to BDNF through this activity-dependent factor, MMP9. So to describe basically a model, let's schematize a dendritic branch with two synaptic inputs. One synaptic input that's a distance delta D from the first synaptic input. We stimulate one of the synaptic inputs and we leave the other one unstimulated. So we know that this matrix metalloproteinase, MMP9, is released in an activity-dependent manner. So upon stimulation of one of these synapses, MMP9 is released at the stimulated synapse, as is indicated here by these yellow squares. So what this results in then is the release, so the stimulation also causes the release of BDNF and proBDNF at the stimulated and the unstimulated synapse. Now, there is slightly more proBDNF at both synapses because, as I told you, especially in development, this proBDNF is slightly more prominent. Upon stimulation, of course, there's the release of neurotransmitter, the opening of voltage-gated and calcium channels, and so we have the influx of postsynaptic calcium in the dendritic branch. And but because of this stimulation of one of the synapses, and we have the activity-dependent release of this MMP9 at the stimulated synapse, we now have tipped off the balance at the stimulated synapse in a different direction in comparison to the unstimulated synapse. Namely, MMP9 ends up converting some of the proBDNF into BDNF, so you now have more BDNF at the stimulated synapse, which eventually would cause potentiation, and you still have more proBDNF at the unstimulated synapse, which will eventually cause this synapse to be depressed. So we basically have asymmetric distribution where there's more BDNF at one synapse and more proBDNF at the other synapse, which causes potentiation in one synapse and depression in the other. And what I've shown you here, the schematic at the bottom, are basically what these traces, so we call these accumulators, namely the calcium, the MMP9, as well as the two neurotrophins, look like at the stimulated and the unstimulated synapse. So you see at the stimulated synapse, we have a very strong release of MMP9. This is the yellow trace, and we don't have that at the unstimulated synapse. And as a result, in the stimulated synapse, the BDNF, which is this green trace, is bigger than proBDNF because of this conversion caused by MMP9, while at the unstimulated synapse, we have slightly more proBDNF than BDNF, and therefore we will have depression of the synapse. Another feature that I haven't actually mentioned to you so far is actually the fact that we have depression of the synapse that depends on distance. So I mentioned here that the unstimulated synapse is delta D away, so some distance away from the stimulated synapse. And as you might imagine, basically this unstimulated synapse is experiencing, through the diffusion of this postsynaptic calcium, is experiencing the activation of the stimulated synapse. But because this calcium obviously does not diffuse infinitely far spatially, there's a certain spatial diffusion space constant, this unstimulated synapse will feel the effects of this calcium only if it's reasonably close to the stimulated synapse. And so therefore, what we show here is if the synapse is nearby, it will experience more of the calcium and therefore it will be depressed more. But if it's far away, it will experience less of this dendritic postsynaptic activation and therefore it'll be depressed less. And so indeed, we can summarize the two features of our activity-dependent plasticity rule that is based on this biophysical mechanism of interacting neurotrophic factors, proBDNF and BDNF, through this activity-dependent released molecule MMP9. So we have two features. So we have the ability of the synapses to become potentiated if they are stimulated, so if they're activated because they release this molecule that will convert the depressing to the potentiated molecule. But because of this postsynaptic diffusion of the calcium, we also have basically the depression of synapses that are reasonably close and not too far away from the stimulated synapses. So if two synapses are stimulated, especially in a correlated manner, they will become potentiated. The synapse that is very close to them but is unstimulated will become strongly depressed. The synapse that is farther away but still under the influence of the postsynaptic calcium will also become depressed, but by a smaller amount. So we can actually quantify this. We can look at how much these synapses will become depressed as a function of how far they are from the stimulated synapse. And so you see here, the farther away they are, so this is the less depressed they become. And of course, the closer they are, so 0 microns, they will become very depressed. And another parameter, of course, that plays a role is the stimulation frequency. So the more you stimulate, the more postsynaptic calcium you have released and the stronger the depression will be. So this is all really great, and we were very excited to be able to kind of conceptualize such a mathematical model that relies on this interaction between these two opposing molecules, one that depresses and one that potentiates synapses. But, you know, this is just one specific mechanism that was identified to operate in the developing hippocampus and was directly implicated in the synaptic clustering that was observed developmentally. However, we wanted to kind of address more generally the problem of synaptic clustering because that was also seen, as I mentioned, in the developing visual cortex and somatosensory cortex. And so, and of course, we're mathematicians, so we wanted to simplify the model to allow us to sort of go beyond long simulations where we can explore all the different parameters. So what we were able to do was to actually find a simplified, or we call this a minimal model, activity-dependent plasticity model, that embodies basically this interaction between BDNF and proBDNF and is of Hebbian nature. So the synaptic change can be described simply as the product of the postsynaptic activity, which is here denoted or represents the spread of postsynaptic calcium, and presynaptic activity, which is basically the activation that the synaptic inputs experience. And then there's a Hebbian term. So this is some offset parameter that I'm not telling you what it is here. It actually depends on a lot of the parameters that we introduced, namely the BDNF and proBDNF binding to the receptors and so on. But what is interesting, sort of from a computational point of view, is how this Hebbian term acts on synaptic change. So basically, this gives depression on the synaptic weights only when you have postsynaptic activity that's proportional to the size of this offset. And actually, this depression that is provided by this Hebbian term, it basically is the form of, it's also can be described as heterosynaptic plasticity, because that's what allows these synapses that are unstimulated and that are far away from the stimulated synapses to become depressed. And so, you know, from now on, we're going to use this so-called minimal model that has this Hebbian term. So it needs pre and postsynaptic activity in order to potentiate the synapse. And this heterosynaptic term that depends on this offset and postsynaptic activity that's actually especially important for the synaptic competition of inputs, and that enables these synapses that are far away to become depressed. So what is nice about this, we have these two features. So let me just reiterate: the correlation-dependent plasticity, which is this Hebbian product, and this distance-dependent competition, which is this heterosynaptic plasticity term. So now we can actually investigate analytically what what we can say, what we can predict about the change in the synaptic weight, in the synaptic efficacy, as a function of these two properties. So one was this correlation. So remember, we have these synaptic inputs that are correlated. So they're experiencing correlated activity and therefore driving the postsynaptic stretch of dendrite. And we also have distance-dependent property of our learning rule through this heterosynaptic competition. And we represent this through the parameter that we call synaptic density. So basically, if on average, synapses are very far away from each other, we have low synaptic density. If on average, they're very close together, locally, we have very high synaptic density. So what we can derive is actually two things. We can first derive an asymptote, which is shown here with this horizontal dashed line, which basically tells us what is the critical correlation above which, independent of the synaptic density, you can never get depression. And we can also derive this other asymptote, or sort of zero curve, which basically tells us when we would have zero change in synaptic efficacy, so basically no potentiation or no depression, so this is a function of combination of the correlation and the synaptic density. And then these blue regions basically denote combinations of correlation and synaptic density where you have depression, or the red regions denote regions where you have potentiation. Let me just note that this is instantaneous change in synaptic efficacy. So if we actually look at how a simple framework with, say, 4 synaptic inputs that we stimulate with, let's say, some kind of correlated activity that follows Poisson statistics, how that evolves in time. So now we simulate a very, very long period in time and look at the evolution of these individual synaptic weights on a given stretch of dendrite. What we consider sort of 3 scenarios. So one scenario is above this asymptote, where we always have potentiation of the synapses. Another, so this is high correlation, independent of density. Another scenario is the star scenario, where we have low correlation and low density. So we're still in a potentiating region, but we're below the asymptote. And then a third scenario where we have low correlation. So same correlation as the star scenario, but we now have high density. So now we're in the blue region, so we actually have depression of the weights. So what I show on the left, let me reiterate again, is instantaneous change in synaptic efficacy. So if we look at these plots that I show on the right, what that means is basically the slope of these trajectories at the starting initial conditions. So for the red cases, I've now colored also the background the same color as the instantaneous synaptic change in this 2-dimensional plot. The slope here denotes that we're kind of upwards, so the change in synaptic efficacy will be positive. And here, in this scenario with the triangle, the slope of these trajectories is negative, so that means that on the instantaneous synaptic change will be negative. But of course, over time, because some of them become depressed, the density actually decreases, so we actually end up moving through this correlation synaptic density landscape. So the plot on the left, let me just say again, it just indicates the slope of these trajectories in time. Okay, so this is, I think, really exciting because it takes us from a very biologically inspired biophysical model that relies on this interaction between BDNF and proBDNF to a phenomenological model that has this Hebbian correlation-based part and this sort of distance-dependent heterosynaptic part that can explain potentiation and depression of synapses.
Yeah.
And yeah.
There's a couple of questions that might be worth answering right now. Someone is asking how the BDNF, BDNF ratio is recovered, like what the timescales of that are, and how that changes the effect. And then someone is asking, maybe answer that one first, and I'll ask the second one right after.
Okay, so the short answer is I don't know the exact numbers, but I think it's something or the order of seconds. So these are sort of time scales. I think the binding itself is reasonably fast, but I think this is a developmental scenario. So you have to think a little bit slower than what you would think about adult dynamics. So in adult cortex, you have spikes, you have dynamics on the order of tens of milliseconds. Here, everything is a little bit slowed down, so it's order of hundreds of milliseconds. And so I think even in the context of BDNF, proBDNF, with how they bind and how they decay, we're thinking of sort of hundreds of milliseconds all the way to the order of a second. And this is important because, yeah, eventually when I start talking about the implications of this, and I really hope I get to that because that's the cool, exciting part, this is really important because also spontaneous activity patterns operate on much slower timescales than sensory evoked patterns in adulthood.
Then maybe a follow-up question regarding the specific areas that you're looking at, and also species. Is this uniform across all animals in all brain regions, or are you looking at a specific place right now? We couldn't see the citations that you're using in the lower left because they're covered by our videos.
Sorry about that. Yeah. So we are motivating, we're extracting all these parameters about how BDNF and proBDNF interact from literature, primarily from rodents. And it's a mixture sort of hippocampus and sensory cortex, and some experiments done in the spinal cord, but all of it is development. So that's why I think slow time scales. But species-wise, most of everything that I'm showing you, so far is almost independent of species, other than the fact that I have kind of constrained the timescales ballpark to be rodent timescales. But so far, the conclusions that I'm making regarding the amount of correlation or synaptic density, these are ranges. And so, yes, a natural question is, what is synaptic density in an animal? So here, my next click was basically some mouse estimate of mouse density in second week of postnatal development, which is just under 0.3 microns, inverse microns.
So there was another question that you're implicitly answering here. Is there a sweet spot of number of synapses per cluster? And I guess you're going to get to that now. Absolutely.
There's a So the factors that determine cluster size. So okay, that's a slightly different question. So I haven't even gotten actually the formation of clusters, so we have to bear with me. I know I'm running out of time, and I have to.
No, we're fine. We've got a lot of time.
A cluster size is determined by several factors. One of them is this diffusion, the spatial scale of the calcium diffusion, and of course of the properties of the synaptic inputs, so the amount of correlation that the synaptic inputs are experiencing. But I'm going to get to clusters in a moment, so I haven't yet gotten to clusters.
Go.
All right, so I'm going to go, and then, I had a natural stopping point, but that's okay. Happy to answer questions now. So there was quite a few questions about species, and this is very exciting because I do want to get to what this model predicts when it comes to clustering in species. So I'm going to just briefly say, because there were questions about species, what does this mean? Like, where are we when we look at data? So indeed, if we look at developing mouse, this is approximately where we are in terms of density. And so indeed, what our model would predict if we are, so we change the amount of correlation. So we kind of take this red line up for a fixed high, so-called high density. So what we see is low synaptic efficacy for synapses that are not very correlated. So here we're in the blue region, and then that saturates. But if we're in a low density region, then we're just flat. We're basically experiencing very little change in the synaptic density. And this looks very similar to what experimentalists found in the mouse visual cortex. So synapses that are basically local, so that are very nearby each other and are in this high density scenario, show indeed if they're experiencing low levels of coactivity, show indeed small change or depression of the synapses, which here is measured as depression in the frequency of activations. But for synapses that are far away, basically, it doesn't matter. It doesn't matter how coactive you are. You basically don't see a change in the synaptic efficacy, which here is measured as a change in the frequency of activation. Okay, so what I really want to get to is the exciting part of what does this mean when we now, take this rule. I mean, this is really nice. This is, mathematical analysis of what this rule does. But the important question is, what does this mean when we actually apply this rule and interpret these spontaneous and allow the rule to interpret these spontaneous activity patterns to give rise to these clusters? Because I started with telling you about clusters, and I actually haven't shown you what it means for clusters. So let's look again at what these spontaneous activity patterns look like. So this is actually a simulation of a spontaneous retinal wave reproduced from a model from the citation below, which maybe you can see. So again, we see these spatially propagating retinal waves. And so now let's think about how we go from these retinal waves, which are basically the natural types of inputs that these synaptic inputs experience in these cortical dendrites, to actual spikes that activate these synaptic inputs. So we have our dendritic stretch, we have our synapses. So how do we generate the spikes? So we used, because we didn't focus on exactly how should this transform be, we used a simple model, namely a linear nonlinear Poisson model. which probably all of you are familiar with. But let me just unravel the individual components. So first of all, the first component is the linear part of the LNP, or linear nonlinear Poisson model. And that is to convert these retinal waves into spikes on these synaptic inputs, we're using linear receptive fields that luckily for us were characterized very recently in a study from Sonja Hofer's group to basically look like Gabor receptive fields. So we all know that individual neurons at the cellular level look like Gabors, and perhaps unsurprisingly, the individual synaptic inputs also have these Gabor-like receptive fields. So they're oriented edges with a positive and a negative component. Great, so now we have this linear filter, so we know what the linear component looks like for each synapse. The second component that we need is the nonlinearity. So we take the linear filter, we take the retinal waves, we pass them through this linear filter. Then we have to take them through a nonlinearity. And so here we just use an exponential nonlinearity. And of course, then we have a Poisson event generator. So we now generate events. I call them events because, as I said, this is development. So they're not really precise spike times, but more like bursty events. And so effectively, you know, we have this red retinal wave that generates a bunch of events here. Then we have the green retinal wave that generates events. And so you can imagine how now these retinal waves are transferred or transformed into spiking or bursty events at the level of the synaptic inputs. Okay, so this is all great. So now let's actually see what this does if we kind of implement it in a big dendritic tree. So this is 150 microns long with individual synaptic inputs. As we said, the individual inputs, which are here color-coded, are very well described by these Gabor filters. So they're oriented edges with a positive and a negative component. So we describe them with two properties. One is the center. So basically, you know, retinotopically, where in the visual world they look at. And the other one is the orientation. And so the color here, so the color on the left of each circle denotes the orientation preference. And here's the color map. And I sometimes also illustrate these synaptic inputs with this positive and negative components shown here with the full and the dashed line. And again, the color indicates the orientation preference. So this like magenta is sort of like 45 degrees, and this yellowish is basically 0 degrees. So let's again summarize what we have. So we have the two important components of our rule. The first one is the correlation-dependent cooperation. So, you know, we have two inputs that by chance, they were positioned randomly on our tree, and by chance happen to have similar orientation preference. So because of that, when a retinal wave passes through the visual field, these two inputs will become co-activated. So they will experience highly correlated activity, and therefore they will become stabilized or potentiated. Of course, we have the second component, which is the distance-dependent competition of the rule. So this is this heterosynaptic term that allows depression of synapses. So here we have another synapse that has almost orthogonal orientation to these two synapses that were highly coactive. And because it experiences this postsynaptic diffusion of calcium through the activation of the first two, over time, it will become depressed. And now we add actually a third component to our model. And the third component that we add is structural plasticity. So what this means is that every time we eliminate the synapse, we allow the birth or the formation of another synapse at a completely random location on the dendritic tree with a completely random orientation preference. And now the process repeats. And so we have this process where we have potentiation and depression, so a learning rule that contains these two properties, as well as structural plasticity that enables some kind of homeostasis so that synaptic inputs don't get completely pruned, or too many inputs don't get added. And so this particular rule eventually reaches a stable state. So let's indeed consider a scenario. We start at postnatal day 0. We apply this retinal wave input with these highly structured spontaneous waves in the retina, and what we get in the end is highly ordered synaptic organization of the synaptic inputs on the dendritic tree. And so let's just, you don't just have to look at this one example and believe me, but let's actually quantify that. So we do this by looking at the cumulative density function as a function of the nearest of the nearest neighbor orientation difference. And so if these inputs were completely randomly organized and were not clustered, what we would get is something that basically goes along the diagonal. But that's not what we get. What we get is a curve that's actually concave down, so it's slightly curved down. So what that means is that you have actually a higher proportion of inputs that have very small orientation difference that are nearby each other. And interestingly, this looks very similar to what was reported in the ferret visual cortex from the lab of David Fitzpatrick. I'm sorry, I don't have the reference here. And so this is wonderful. This means that we have clustering now in terms of orientation. But we can also, as was done in the experimental data, we can look at clustering in a similar way as was done in the spontaneous activity experiments in development. Namely, we can look at how correlated or how coactive individual inputs are as a function of their distance. And so in the full line here, we show results from a model. So what we see indeed is clustering, namely that synapses that are nearby each other, that have a small pairwise distance between them, experience high correlation, and as distance decays, they experience less and less correlation, which is the signature of the clustering. And this very nicely matches to what was characterized experimentally in the ferret visual cortex. So these are similar experiments, except at the top they showed the drifting gratings, and at the bottom they just didn't show any visual inputs. So completely in the dark environment without any visual stimuli, they just measured these correlations. Cool. So this is great. So we have orientation clustering in the ferret, but some of you might be thinking, or you know that actually the mouse doesn't show that. So a different way to show this clustering is to actually investigate, to plot the orientation difference as a function of the pairwise distance. So what we show here, we show a lower than chance orientation difference for synaptic inputs that are nearby each other, while as the synaptic inputs get far away from each other, their orientation difference approaches chance, which is 45 degrees. So this is the results for the ferret that match this clustering that I mentioned in the ferret. But in the mouse, this is not the case. So this study that I mentioned from Sonja Hofer's lab, but also some earlier work, has shown actually that the synaptic inputs in the mouse are not orientation clustered. Namely, as a function of distance, the orientation preference between these pairs of synaptic inputs is actually literally on top of what is predicted by chance. So we had an idea for what might actually underlie this difference in the species. And our idea actually, so this is one of the most important conclusions that we have from the work today, is the idea is that it has to do with the relative amplification factor in the two different animals. So basically the sizes of these two different cortices are different. So the ferret cortex is about 30 times bigger than the mouse cortex, but the density of the neurons is about the same. So that means that the amplification factor in the ferret is much, much bigger than in the mouse. So if you are so what we show here in blue lines are actually ISO curves that denote effectively retinotopy space in azimuth and elevation in the visual world. And if you are some kind of reference, if you look at some reference pyramidal neurons shown here with the gray triangle, either in the ferret or in the mouse cortex, and you look at what types of synaptic inputs this reference pyramidal neuron pulls from the environment, you see that in the ferret, this neuron pulls inputs from basically neurons that are nearby retinotopically, and in the mouse, it pulls inputs from neurons that are very far away in the visual world. So we can illustrate this by effectively superimposing the centers of these receptive fields in the ferret. So these are a bunch of gabors, which basically indicate the receptive fields of these neurons in the ferret, and then below we show that in the mouse. So in the mouse, they're much more dispersed because we have a much smaller amplification factor in the mouse. And we actually take this factor of how dispersed these receptive fields are from data. So we use data from ferret and we use data from mouse that is shown here at the bottom. So you really see this much bigger dispersion in the mouse. And so indeed, what our activity-dependent learning rule predicts is that in the ferret, because of this small dispersion, so namely the bunching up effectively of these centers of the receptive fields, the effect, what we get is orientation clustering, the result that I showed you. But what we see in the mouse is a different type of clustering. So it's clustering with respect to a different feature. And the different feature in this case is actually overlap of receptive fields. So you see here a bunch of receptive fields that organize themselves in such a way that they're clearly not orientation clustered, right? So all of these gabors effectively kind of span the entire wheel of orientation preference, but they all have very strong overlap because the positive parts of the gabors, which are indicated here by the full line of these ellipses, basically are all on top of each other. So we have overlap clustering in the mouse and orientation clustering in the ferret. In fact, overlap clustering is also a subset of orientation clustering. So of course, the ferret will also show overlap clustering even stronger than the mouse, because of course these positive parts of the receptive field here will overlap as well. So that's what we show here at the bottom. So we show the simulation of the ferrets. So again, we look at the overlap as a function of pairwise distance. So we see that's very strong for synapses that are nearby each other and decays that the synapses go far away from each other. For the mouse, we again see a significant relationship. So again, neurons, pairs of synapses that are nearby each other show large amount of overlap, shown here at the bottom. And this looks very similar to, or this pattern resembles what is seen also in the data. So I think I'm going to run out of time, but what I have shown you in this first part, just to summarize very briefly, is the emergence of these two types of local clustering on these small dendrites. One is orientation clustering in the ferret, and a combination of overlap and orientation clustering, orientation and overlap clustering in the ferret, but only overlap clustering in the mouse. And so we think that the main reason that basically drives these differences is what is the amplification factor? So how spread out are the inputs that a given neuron receives in its environment? So Tim, tell me, do I have time to show you a few more minutes, or is
we started a little bit late, so you have an easy 10 minutes to go at least.
Okay. Okay. Maybe I won't take the whole time, but I'll try to take 5 minutes to at least show you the next cool result that we have. So because
there's no stress.
Okay. I don't want people to have to leave, but I guess if they have to leave,
they can just log out.
Okay. They can also stream the rest if they have to be somewhere. Great.
Yeah.
Okay. So what I wanted to show you next is, so we're very excited about this because it tells us how synaptic inputs become organized locally. It's clustering. It's how I motivated basically the approach today. But this allows us to actually ask questions beyond this local organization and actually go beyond local to global and ask how synaptic inputs might also be organized globally on the entire dendritic tree. So now we blow up the problem. So instead of just a small dendritic stretch, we look at an entire morphology of a pyramidal neuron taken from the Allen Institute database, and we basically apply the same kind of approach, the same kind of plasticity rule that interprets these spontaneous activity patterns and uses them to organize synaptic inputs on the tree. Oops. Okay. The additional element that we add in this case is a way for these clusters that we now know are formed in parts of the dendritic tree to talk to each other, to communicate to each other and to the soma. And we do that by effectively using the mechanism of back-propagating action potential. So in other words, if there's a very strong signal somewhere on any part of the dendritic tree of this neuron, this signal gets integrated and the soma generates then a powerful back-propagating action potential studied by labs for many years now, that ends up basically retrograding the signal back to the inputs at the different portions of the dendritic tree far away from the soma. So here's this example. So we have the generation of this somatic signal, somatic activation that integrates these inputs. And then once it crosses threshold, we have the generation of this back propagating action potential that goes and reaches the inputs far away. Of course, these BAPs are not super powerful. They attenuate as they get far away from the soma, and so there's certain sort of spatial decay constants as they get away from the soma to the tips of the dendrites. And so we basically applied our learning rule with the spontaneous waves and this addition of backpropagating action potentials to study what type of organization now emerges in these entire dendritic morphologies. And what we found was actually global order in the dendritic tree. So here's an example, and I think if you stare at this for long enough, you will hopefully realize that the synaptic inputs that are nearby the soma tend to share similar orientation preference, and the inputs that are far away are still locally clustered, but are sort of independent and have orientation preference that's independent of the soma. So one way to quantify this is to actually, so what we did is we divided up the inputs into what we call proximal, that are further away than 150 microns from the soma, and opposite, that are closer than 150 microns from the soma, and distal, that are far away, so more than 150 microns from the soma. And then again, we plotted the cumulative density function for the circular dispersion. So this is basically the difference in orientation preference between a pair of neurons. So I've tried to indicate it here basically. So here we have one synaptic input that kind of prefers this orientation. Here we have another synaptic input that kind of prefers, almost orthogonal orientation. And so the circular dispersion is basically the difference between these two angles. And so what we see is for inputs that are far away, as I told you, the relationship is basically, a flat line. So what that means is that these inputs are almost, if you compare them to the preference at the soma, they're almost, their orientation preference is almost unrelated to the soma. But if you look at inputs that are nearby the soma, what you see is that there's a very strong probability that they will actually share the same orientation preference to the soma. Namely, they will have a very small circular dispersion. So the angle difference, the orientation preference, will be very similar to the soma. So this is a quantification of the global orientation gradient that we get of how these synaptic inputs organize on the entire dendritic tree. And what was curious to us was that this actually looked very similar to a different type of classification of synaptic inputs that the Fitzpatrick lab generated in the ferret data. Namely, they divided the data into clustered and heterogeneous branches. So they didn't look at whether they were proximal or distal. They just looked at whether the branches had a lot of clusters or whether they were they had fewer clusters and looked more heterogeneous. And so in some sense, this is just a kind of correlative analysis, but we would suggest that maybe this clustering or heterogeneity of synaptic inputs that they found in the data could be related to how far these synaptic inputs are on the dendritic tree, and therefore is controlled by the strength of the BAP. So the closer you are to the soma, the stronger you experience the BAP, so you're more strongly entrained to acquire the similar orientation preference as the soma, and the far away you are, you experience the BAP less strongly, and therefore you will acquire orientation preference that's independent of the soma. But in the mouse, that is not the case. And so this is just the summary where we actually changed the attenuation factor of the BAP. This is, basically just telling us that, maybe unsurprising, I mean, we don't have this local orientation clustering in the mouse, so why should we expect global orientation clustering? So this is perhaps unsurprising. This is a general summary for the ferret, and now we see it for the mouse, that indeed it's very close to chance. But what is interesting is in the mouse, we get a different type of global organization of synaptic input. So remember, in the ferret, locally, we have orientation preference, and in the mouse, locally, we have overlap clustering. So in the ferret, they're clustered by orientation. In the mouse, they're clustered by overlap. In the ferret, we got that globally, they are organized away from the soma with this orientation preference. So the closer you are to the soma, the similar preference you share to the soma. And now what we get in the mouse, as a result of this tendency to actually get this overlap clustering locally, we actually have a global organization by a different feature than orientation. And this different feature here is visual topography. So what I mean by that is effectively synaptic inputs that are nearby the soma end up coding for positions in the visual world that are nearby, so that as the soma, and synaptic inputs that are far away code retinotopically for positions that are far away from the positions coded by the soma. And so we can look at basically, so the strength of the BAP changes. So if we have very sharp decay of the BAP or very sort of strong BAP, we can look at, it's very difficult to see this effect if you look at an individual instance of this global order, but if you look at summaries, you really see this effect. So here the color just indicates in the visual world, so how similar is your topographic preference to the soma. So the small numbers here denote that you really like to see things that are nearby in the visual field, and the farther away you'd like to see things that are far away in the visual field. So we can quantify this similar to what we did for the orientation for the ferret. And so indeed, if we change the attenuation of the BAP, we see indeed that we have this this receptive field offset that really changes as a function of distance to the soma. So a synapse that is close to the soma will have a smaller receptive field offset, meaning that they really look at similar points in the visual space, and the farther away you are from the soma as a synapse, you really look at different portion in the visual space. And so this is very nicely matched, or very nicely sort of predicts data from the lab from Sonja Hofer that identified this very nice emergence of visual topography globally of these dendritic trees in pyramidal neurons in the mouse. And I think I'm going to end here, but my last parts of the talk were actually about the emergence of computation and what this might be interesting for. So maybe I just leave you with this thought as the end, and that is that the reason. So what might be a computational advantage of having this organization of these synaptic inputs in this topographic way, that nearby inputs to the soma code for nearby inputs in visual space? Well, it might be good for detection of edges in natural images. So, for example, if this red box is the preference of the receptive field of the soma, and sort of these green ones are synaptic inputs on the dendritic tree. So the fact that, I have to explain the other concept of collinearity here. Okay. Sorry, I'll explain this. So this kind of organization that I explained to you that emerges at the kind of globally at the level of the mouse, of the dendritic tree of the mouse, gives rise to another effect, and that is the emergence of collinearity computation. So to really visualize this, we look at basically the receptive field preference of the soma. So in this case, it's 45 degrees. So this was actually a computation that was proposed by Caruso et al. In Sonja Hofer's group. And they define these two types of spaces, what they call orthogonal and coaxial space. So coaxial space is one that's basically co-aligned with the orientation preference of the soma. And orthogonal is basically the orthogonal space. And what they found is that there's overrepresentation of synaptic inputs that basically are in this coaxial space that actually share similar orientation to the soma, and there's less of prevalence of these synaptic inputs that are orthogonal to the soma but are present in this coaxial space. And this is exactly what is important for the detection of this collinearity. So these basically extended edges that exist in natural images. So this would be like, as I said earlier, the location of the soma, and this would be the location of the synaptic inputs that then basically help the neuron to effectively detect that this is an edge, an extended edge. And what we find actually slightly different from the data, we can actually look at the prevalence of these in the orthogonal space. So here we focus on the orthogonal space, and so here the model is shown with the bars. We actually see an overrepresentation of synaptic inputs that are orthogonally tuned to the soma in this orthogonal space. And we think that that's actually important for the detection of these co-circular features that exist in some natural images, where basically you have two orthogonal portions of like a circular contour that are basically tangent to this circular contour. And so We think that they don't actually find it in the data. Of course, this is in vivo data, and so it's a bit complicated. But in our model, we find this prevalence, and so we think that we have this nice kind of prevalence of these two different types of computations: the detection of these collinear and the detection of these co-circular features. Okay, so this is my last slide. I want to summarize that basically two results that I've shown you. I mean, I spent the probably too long of the first part of my talk motivating the biophysical model based on this BDNF proBDNF interaction that was identified developmentally in the hippocampus to be responsible for clustering. But then what I showed you later was actually the product of a mathematical model that was derived directly from this proBDNF BDNF biophysical model. That basically naturally extended all the time scales that we put inherent in our BDNF proBDNF model, and enabled us to explain two different types of organization. First, local organization on small regions of dendritic trees, which in the ferret gave rise to clustering in terms of orientation, and in the mouse, clustering in terms of overlap. But also global organization, once we introduced this global homogenizing signal in the form of a back-propagating action potential, that gave us the emergence of orientation preference gradients. So the farther away from the soma you get, the less similarly tuned you are to the soma. And also this retinotopic or sort of visual topography gradient, whereby synapses nearby the soma tend to prefer sort of code for similar locations in the visual space. So this retinotopic gradient. And this is it. Sorry to go over a bit. I hope people are still here and have questions. Just want to thank my group, and especially my student Jan, who did a majority of this work. And I especially want to thank Christian Lohman, who is my collaborator from the Netherlands Institute for Neuroscience in Amsterdam. We collaborate on many other projects together. In this particular project, he's not a direct collaborator, but really some of the work that I described is very directly inspired by some of the recent experiments that they have done. And of course, my funding sources, and thank you very much for your attention and for sticking around for so long. Happy to answer more questions. I think hopefully there are many more questions now.
Thank you, Julijana. There's no clapping function other than graphic clapping function, so it's always a bit of a letdown, but there are still quite a few questions.
Good.
So some of them are more detailed, others are more global. We actually had a question from someone who seems to be joining us from outside of neuroscience a little bit. So someone asked what the long-term impact of your research will be. I thought it was kind of cool that we had someone coming in from further out, so asking actual fundamental questions. Some of the other questions were more with regard to how your homeostatic plasticity is implemented and how you sprinkle in these new synapses. So if I understand correctly, you have a net loss of the synapses always, that also allows you to create this competitive effect between near and far synapses, and then you sprinkle in new synapses just to compensate for that net loss. Is that correct?
That is correct. So synapses are reinforced if they experience correlated activity, and synapses are depressed if they are nearby active synapses, but they're not active themselves. And they're, of course, they're not active because they have different anti-correlated activity, maybe with the activated synapses. And then every time a synapse is depressed, a new one is born. And so in the end, we actually reach a dynamic balance. So the synapses do not remain static. We always have some baseline level of turnover. So the orientation preference basically remains. The orientation clustering that we get remains, but we have some kind of a dynamic balance where some fraction of synapses continue being turned over.
Get cycled over. Sorry, I'm trying to read and listen at the same time. But in that model, I think a lot of it depends on how you design your spontaneous activity or your retinal wave or whatever have you, right? So is that true, what I just said? I guess, and then do we use the same retinal waves for ferret and mouse? And by how much can you affect, what came first, the receptive field or the retinal wave there? Okay,
so there's really several questions here. So the first one is about the spontaneous activity and sort of how sensitive is the model to the spontaneous activity patterns. And I will say not sensitive at all, to the point where what we just need are long time scales. I mean, I mentioned several times now, we're considering development, and so we're, the time scales that we sort of try to extract and model from existing data about the BDNF, proBDNF interactions and so on are all slightly slower than what we would expect later on. And so the developmental patterns are also slower. So we have here correlation time scales on the order of several hundred of milliseconds. And so as long as you have this slowness, and maybe the slowness is not even so important, but the match between the time scales of the activity and the time scales of the learning rules, you're fine. You're going to get the emergence of some kind of structure. And whether spontaneous events are similar between mouse and ferret, I mean, there are certain differences. Ferret, actually, eyes are closed for much longer than in the mouse, and people have indeed measured retinal wave properties. I think, so this is currently a preprint, and this paper is being revised, and at some point we were asked about using maybe properties of retinal waves from ferret, and we were able to find a model and modify the properties from the ferret. There's of course differences, but the correlation timescales, as long as they're matched to those of the activity patterns, basically the properties of the results of what we showed basically remain. And then I now forget, but you were asking me, so one was about the spontaneous activity.
What came first, receptive field or receptive wave?
Yeah, that's a great question. So, we used the receptive fields. They were identified in the adult mouse. And so we used them, through this linear nonlinear Poisson model to basically go from, the spontaneous activity to the basically spikes. And then we can use our plasticity rule. But of course, they were identified in the adult mouse. And so the question is, can we really assume that these individual synaptic inputs are basically orientation tuned? We know actually from work from Tomáš-Flogel's group published, I think, early 2010s, that indeed at the cellular level, the neurons are orientation selective at eye opening. So it is not completely unfeasible that maybe they acquire this orientation preference just before eye opening, but this is exactly the period that we're modeling. So there is this, at least at eye opening, we have this orientation preference. But we also did some work where we actually looked at simultaneously the emergence of orientation selectivity through refinements from the LGN. So we basically have center surround inputs from the LGN, so on and off, and simultaneous to the sorting that we have in cortex in terms of these orientation-tuned inputs in clusters, we also simultaneously had the emergence of orientation selectivity through this convergence from LGN to cortex. And, at least sort of to the best of our knowledge, I mean, we didn't try like super hard, but this seemed to also work. So we can have a model where we can have them emerge simultaneously to the organization, to their self-organization in cortex.
So then two more mechanistic questions. One is about interaction of voltage, internal voltage in a multi-compartment model with your proBDNF, BDNF model. And the other one is about the very different internal mechanisms of apical versus basal dendrites. So what about NMDA plateaus in the far tuft?
Okay. The first question is, our model is not multi-compartment, so it's basically we extract the morphology, but in our model the only thing that matters is really like where the synaptic inputs are distance-wise from each other and from the soma. And so we have a very simple mechanism by which the synaptic inputs are integrated to give rise to this BAP that then propagates outward from the soma. So we don't actually have a multi-compartment model with many ion channels, and so we don't actually have voltage that we have to worry about. I think this is an excellent question. Otherwise, I mean, we are thinking potentially about making the model kind of more realistic to also incorporate these aspects of the ion channels and how these different compartments could interact and what they could bring to the system, and how this could interact with these activity-dependent plasticity mechanisms. But at the moment, this is not something we have addressed. And then the second question was about the differences between apical and basal dendrites. Again, In our model, the only difference between them at the moment is how far away they are from the soma. So there are no other differences in terms of NMDA, or which I know actually just before the seminar I had a great meeting with somebody from ISD about differences between apical and basal dendrites, and I know there's a lot of literature out there, and this is something that we really care about and plan to incorporate in the future. But at the moment, the only difference is how far they are from the soma. So in that sense, basal are close, apical far away, and in that sense, proximal, apical, and basal would be the same, which I know is bad to say, but at least in our model, it's distance that matters.
Okay, and then maybe to finish, and maybe more zoomed out question: do you think the different kinds of clustering in mice and ferrets have functional consequences for how the species see? And do you think humans are more like ferrets or more like mice?
That's an awesome question. I think absolutely that there are functional consequences, and so I try to get that a little bit at kind of this part. I mean, this was primarily focusing on the kinds of clustering or organization, global and local, that we found in the mouse. So I absolutely think that there are implications for some implications for what this clustering could really mean. Yeah, I mean, we're brainstorming. Maybe one could do some hypothetical psychophysical experiments on our synthetic neurons and ask what these differences could be. But at this point, it's very hypothetical. But I think it's a super relevant question to go beyond, yes, the activity-dependent organization, which is interesting, but to actually what these things mean. And with regards to, are we similar to
mice or ferrets?
I don't know. Probably we're more similar to mice. I mean, I don't know if I have a spare slide. We tried to go a little bit, maybe just focus on the top slide here. We tried to go a little bit beyond mouse and ferret, and we actually tried to infer like this amplification factor that I thought is the important thing that determines the differences between mouse and ferret, and we tried to extract. So I told you that it's the receptive field center spread, so how far a given neuron pulls from other neurons around it. The other thing that we think is important is also the receptive field diameter. And so we try to kind of infer just these parameters very broadly from literature for different species. So for example, rabbit, cat, macaque. And we kind of try to put them in this landscape and try to kind of make predictions. So ferret would be here. So green here. I think maybe you can use the color scheme at the bottom. Green is orientation clustering. Purple is mouse. Mouse is here. Would be overlap clustering, and so we kind of have these data which are all really close to each other for rabbit, cat, and macaque. Recently, just last year, or maybe even earlier this year, there was a paper from macaque that actually shows indeed you have orientation clustering in macaque. So macaque is like ferret, so probably we are like ferret too. We have orientation clustering, and then we kind of speculate what could be the case, for rabbit and cat. So, yeah.
Cool. Thank you very much. I think that's enough for questioning. Thank you for a lovely talk, Julijana.
Thank you. I encourage everybody to, feel free to email me or get in touch if you have other questions. Yeah.
Great. Okay. With that, thanks to the audience. Thank you. And I'll see you on the other side on Zoom.
Yeah.
All right. Goodbye, everyone.